I thank Auro Siksha for giving me this opportunity to conduct this webinar. I hope this is very useful for everyone who's watching. So, my colleague Dr. Priyanka first touched upon the anatomy of the orbit and extraocular structures. So basically, the skull is like a house. So the orbit is two rooms inside the house, and the eyeball and other ocular structures are contents of those rooms. So I am going to talk about the anatomy of the eye proper. So the topics for discussion is going to be in a systematic order. As you can see in this image, the eye can be compared to a camera. So similar to a camera, the eye has a lens. Light rays fall on the eyes, they get refracted, they fall on the retina, which captures the image. Similar to how a camera captures images, our eye captures images, records videos on a daily basis, which we take it for granted. But let's take a look at the anatomy of eye, because understanding anatomy is very, very important. So the first, I'll be talking about the human eyeball and giving you an overview about it, and then the anatomy of the eye from anterior to posterior. It is always you should always study anatomy in a systematic fashion. So first we'll start with the anterior most structure, which is the cornea. Then we'll go with the sclera, the uveal tract, the lens, the vitreous, the retina, and finally the optic nerve. So the human eyeball. So generally we think whenever we think of an eyeball, we think it's like a globe, but it's not entirely true, because as you can see here, it's not a perfect globe. You have a smaller sphere here, which is attached to a much larger sphere. So let's take a look at in detail what that is. So before that, let's look at the few important dimensions which you need to remember. The first one, the most important, is the anterior posterior diameter, which is around 24 millimeter. And the horizontal diameter is 23.5 millimeter, vertical diameter, which is 23 millimeter, volume is 6.5 ml, the weight is 7 grams. Even if you forget all these measurements, the one thing which you cannot afford to forget is the first one, which is the anterior posterior diameter from here to here. So this measurement is important because usually the anterior posterior diameter, otherwise called the AP diameter, ranges from 23 to 25 millimeter. So coming to the coats of the eyeball. See, the eyeball is basically, you can imagine it to be like an orange, which has layers, and we're going to peel each one and we're going to study it. So the outer protective layer, which is the outermost one, is called the fibrous layer, which is composed of the sclera, the cornea, and the limbus. The middle vascular layer, which is uvea, which is composed of the iris, ciliary body, choroid. And the innermost important layer is the neural layer, also known as the retina. So here are the three layers. As promised, I have peeled all these layers to show it's simplified for you. So the outermost one is the sclera, the second layer is the choroid, and the innermost one is the retina. So coming to the structure of an eyeball, so you would have heard terms like anterior segment, posterior segment, anterior chamber, and all these other things which may confuse certain people. So I have taken my best effort to simplify these things. So this is a schematic diagram of a human eyeball, and this is the anterior end, and while this is the posterior end. So this structure right here, you see, is called the anterior chamber. It starts from the cornea up to the iris. Next one is the posterior chamber. It comprises of structures from the iris to the lens. So the anterior segment is nothing but the first two things which we just saw. It is the anterior chamber plus the posterior chamber. Together, these two structures are called the anterior segment. And finally, we have the structures behind the lens. It's called the posterior segment, which comprises of the vitreous, the retina, and the optic disc. So to summarize what we just saw, the eyeball is divided into the anterior and posterior segment. The anterior segment is composed of anterior chamber and posterior chamber, while the anterior chamber comprises of these structures which we have previously just seen. So first, as I said, we're going to go from the anterior end to the posterior end. So first is going to be the anterior chamber structures, which I'm going to talk about three topics here: the cornea, sclera, and the limbus. So the cornea. So cornea is the most obvious anterior structure which we see whenever we look at somebody's eyes. It forms the anterior 1/6 of the outer fibrous layer of the human eyeball. The cardinal features or the important features are it's transparent and it is avascular. It is transparent because only if it is transparent, the light rays can pass easily without hindrance, and it is avascular. That means it does not have any blood supply. And that factor, that being it avascular, is a very significant factor responsible for being transparent. So it is the first refractive medium of the eye, also the most important refractive structure in the eye. The refractive power of the cornea is 45 diopters. So the total refractive power of the eye is 60 diopters. So 45 out of 60. So this implies that the cornea is the most important refractive structure in the eye. So what happens what about the remaining 15 diopters? I guess most of you can easily guess that, but let's see in the subsequent slides. So coming to the layers of the cornea, it has 5 layers from the anterior to the posterior. It goes as follows: the anterior epithelium, the Bowman's membrane, corneal stroma, Decimet's membrane, and endothelium. An easy mnemonic to remember these 5 layers, I think most of you will already be aware of it. It is A, B, C, D, E, which when the first letters of all these words makes this mnemonic A, B, C, D, and E. So the first structure is the anterior epithelium. It is the outermost layer composed of 5 to 6 layers of cells. It provides a smooth refractive surface. It is capable of quick wound healing and regeneration. So this here you see is the normal cornea. This does not look like a normal cornea, is it? Because you can see a very clear epithelial defect. But one good thing about an epithelial defect is that it heals quicker with good prognosis. So whenever a person comes with any foreign body injury or any other trauma history, always stain the cornea with a fluorescent stain and see with a blue filter, and you can easily pick up these epithelial defects. The second layer is the Bowman's layer, which is composed of collagen fibers. It cannot regenerate if damaged, unlike the epithelium. So what happens if the Bowman's layer is injured? This is what happens. Once eroded, it will result in the formation of superficial scars. Coming to the third layer, the bulkiest part of the cornea, the corneal stroma. It's made of type 1 collagen fibers. It is composed of 90% of cornea. It provides transparency, strength, and elasticity to cornea. And the fourth structure, which we see here, the green one here, is the Decimet's membrane. It's made up of type 4 collagen. It, however, can regenerate. The main function of this Decimet membrane is it provides rigidity and stiffness to the cornea. And the last and the final structure is the corneal endothelium. It is the innermost layer of the cornea. It is composed of a single layer of cells. A normal endothelial cell count is around 3000 cells per millimeter cube. What is the function of these endothelial cells? Because it has active pump mechanism. That means it keeps the cornea dehydrated. So as you can see from the stroma, these endothelial cells continuously pumps out excess water or fluid from the stroma out of the cells, like similar to any hand water pump or something. So what happens when this endothelial layer is damaged? This is what happens. As you can see, the cornea is not transparent. It has become hazy. It is not avascular. You can see blood vessels are penetrated into it. So this does not look normal, right? Yes, because this is called corneal decompensation. Loss of endothelial cells more than 75% will result in this condition called corneal decompensation. If left untreated, it has a very, very poor prognosis. And the only thing we can do is corneal transplantation.
Dr. Aravind, a quick comment.
Yes.
We're just receiving some comments about Dua's layer. So you just want to maybe just quickly go back and mention the 6th layer? Yeah.
Okay, so there is another layer called the Dua's layer. However, it's still under the debate, but it has been proven by an Indian scientist living in the UK. That's called Dua's layer, which is considered to be the 6th layer of cornea. It's considered to be a pre-Decimet's, that is in front of the Decimet's. You can get another layer called the Dua's layer, which is considered to be the strongest layer in the cornea. It was recently discovered by an Indian ophthalmologist living in the UK. I hope that is clear. So coming to the sclera, it forms the posterior 5/6 part of the external fibrous layer. It is commonly regarded to as the white of the eye. The white structure you see is the sclera. The outer surface is covered by a thin fibrous capsule called the Tenon's capsule. So the thickness of sclera is variable. In adults, it's much more thicker than in children. In males, it's much more thicker than in females. So the layers of the sclera. The sclera consists of following three layers, again from anterior to posterior. So the first one is the episcleral tissue, which has vascularized layer because it has vessels. Then the sclera proper, which is avascular structure. And finally, we have the lamina fusca. So coming to the limbus is nothing but the transitional zone between the cornea and the sclera. The bulbar conjunctiva gets terminated at the limbus, as you can see around this part. So the limbus, the significance is it contains something called the limbal stem cells, which have ability to differentiate. And a limbal stem cell deficiency disorder will result in the formation of a pterygium. A pterygium is a wing-shaped tissue or growth of the conjunctiva encroaching upon the cornea, and it can result in any visual problems or sometimes mostly cosmetic problems. So coming to our first question, which layer of the cornea has the ability to regenerate? The poll will be pulled up now. So you can, so which layer of the cornea has the ability to regenerate? So is it the Bowman's? Is it the endothelium? Is it the corneal stroma? Or is it the epithelium? Yeah, 75% have voted correctly. It's the corneal epithelium. And coming to the second question. Yes. What is the primary function of the corneal endothelium? And this, you will also have a poll for this also. Okay, there has been mixed responses, but the majority has voted right. It pumps excess fluid out of the corneal stroma. You're right. So let's move on to the posterior chamber. So here I'm going to talk about the structures like the iris, ciliary body, choroid, and the lens. So the uveal tract, what is uveal tract basically? It is divided into three parts. The iris, ciliary body, and the choroid together is called the uveal tract. So the iris is the anterior most part of the uveal tract. It is a circular disc corresponding to a diaphragm of a camera. It has a central opening called the pupil, which regulates the amount of light that enters. It has two muscles. It is a constrictor and dilator muscles. The significance of this is drugs like pilocarpine, which is an anti-glaucoma drug. It acts on the constrictor muscles, while tropicamide and atropine are dilators, and they act on the dilator muscles. So what happens to the iris and the pupil when exposed to light? So in bright light, the iris contracts and the pupil will constricts. In dim lights, as you can see, the pupil dilates so that more light can enter in. So ciliary body, coming to ciliary body, it contains two structures, ciliary muscles and ciliary processes. The ciliary muscles take part in accommodation. What is accommodation? It is nothing but changing focus at different distances by changing shape of the lens. So similar to how a camera can change its focus, how we take images at different distances, that's what the function of a ciliary body does. So drugs like cyclopentolate, which you all commonly use in your clinics, it relaxes these ciliary muscles and prevents accommodation. This is used mainly in children during refraction, especially less than 18 years or less than 15 years, because they have very high accommodative power, and that will give you a wrong value in refraction. And using cyclopentolate will help you in abolishing accommodation. So this is the image of a ciliary body, which as you can see is zoomed here. And this is a ciliary body with a ciliary muscles, ciliary processes, and zonules, which is attached to the lens here. And this will be covered in detail in the anatomy of lens. So just to touch upon the aqueous humor pathway, which is more of a physiology topic, but aqueous humor is present in both in the posterior chamber and the anterior chamber. It is a fluid which is produced by the ciliary processes. As you can see here, it travels along the posterior chamber. It enters the anterior chamber through the pupil. It goes along the periphery and it drains in this angle, which is the iridocorneal angle, and it drains out through the trabecular meshwork and into the episcleral veins, and it comes out. So any problem in the drainage of aqueous humor will result in glaucoma. And final structure of the uveal tract is the choroid, which is the posterior most part of the vascular coat. It extends from the optic disc up to the ora serrata. Its inner surface is smooth, brown, and lies in contact with the retina. So this is an image showing a choroidal detachment. So that is just another clinical thing which you might need to know. So coming to the human lens, it is a transparent biconvex crystalline structure of the diameter of 9 to 10 millimeter. The thickness of the lens varies with age. It's around 3.5 millimeter at birth to 5 millimeter when you grow up. So an elderly gentleman or a woman will have a much harder, much thicker lens because the thickness increases. The refractive power of the lens is 15 diopter. So the total refractive power of the eye is 60. I hope you all remember, the remaining 45 diopter is contributed by the cornea, and the remaining 15, which we did not discuss there, is here. It is the refractive power of the lens, which contributes the remaining. And the lens is attached to the ciliary body by the zonules of Zinn. So here you can see a very nice image showing the parts of the lens compared to another very nice image on your right. So when summer is coming, I'm sure all of you are eager to eat this fruit, king of all fruits, the mango. And the part of the lens can be compared to the parts of the mango. As you can see, the lens has three parts: the inner nucleus, the outer cortex, and the external fiber, the capsule, which covers the lens. And you can see, similar to the lens, you have, this is the nucleus, this is the cortex, and this is the capsule. So to know about the parts of the lens, because you have to know that the lens has a nucleus part and a cortical part. So when the nucleus part gets cataractus, it is called the nuclear cataract, and the cortical, the outer part, when it gets opacified, it's called the cortical cataract. These are two separate entities which you need to differentiate. So the lens, as I previously said, they're attached to the ciliary body by zonules of Zinn. So what is the significance of that? As I said previously, when the lens can change its shape whenever we see objects at different distances. For example, when you see a distance object like a tree, the ciliary muscles are relaxed, the zonules are taut, and the lens a little bit more flattered. So such that the rays can focus on the retina. When you see something, a close object like a phone, or when you read newspaper or a book, and when it's close by, the light rays from the object is much more diverged. So you need much more higher refractive power to converge the light rays back into the retina. So when you bring an object closer, what happens is the ciliary muscles contracts, the zonules are relaxed, the lens becomes much more rounded. And whenever the lens becomes rounded, its refractive index power increases such that the rays still focus on the retina. And what happens when this is problematic? It results in something called presbyopia, which usually occurs after 40 years. And problems, it results, the patient complains he cannot see phones, or he'll have difficulty in reading. So coming to the third question, ciliary muscles are responsible for aqueous secretion. This is a true or false question, and it has only two options. So I think you can bring up the poll now. Okay, we have 63% has said true. Okay, fine. It is actually, the answer is false, because the ciliary muscles are not responsible for aqueous secretion. The ciliary muscles are responsible for accommodation. Aqueous secretion is done by the ciliary processes. So coming to the fourth question, the lens is attached to the ciliary body by which of the following? You can bring up the fourth poll now. Yes, so the lens is attached to the ciliary body by which of the following structures? Is it the iris, the lens capsule, the zonules of Zinn, or vitreous? Yes, 83% have got it right. It is the zonules of Zinn. So coming to the final posterior segment, now we are going to talk about these structures here, which is the vitreous humor, the retina, and the optic disc. So what is the vitreous humor? It is an inert, transparent, jelly-like structure that fills the posterior segment of the eyeball. Its functions are, it's a transparent medium for the light rays to pass. It has a mechanical stabilization of the globe. It provides a pathway for the nutrients to reach the lens here and the retina. So coming to the retina, it is the innermost layer of the eyeball. It is very thin and transparent. As you can see, the retina is actually transparent. So when you look at the fundus and you see something like this, the orange or the red or the yellowish glow, it is not actually the retina which contributes to the color. It is the underlying choroid. The retina per se is transparent. It does not have any color. It extends from the optic disc to the ora serrata. It contains photoreceptor, which has rods and cones that help us see. The retina can be compared to the film of a camera. The functions of the retina are it records the image and transmits to the brain through the optic nerve fibers. So what are the parts of the retina? It has three parts. It has an optic disc, which is otherwise called the blind spot, point from where it is a point from where the optic nerve leaves the eyeball. Rods and cones are actually absent in this place, hence no vision. That's why this part is called the blind spot. The second part is the macula, which is responsible for the central vision, and center of the macula is called the fovea. And fovea is the part where you have the maximum concentration of cones and is most sensitive to light. And finally, you have the ora serrata. It is a junction of the periphery of the retina and ciliary body. The retina starts from here and it ends with here. And the margin or the periphery of the retina is called the ora serrata. So there are four types of cells in the retina, which I'm going to teach you in a systematic fashion. So first one is the retinal pigment epithelium. So this structure, you need to imagine that this one is the anterior part. Here is the posterior part. That means you have the cornea, the lens, and the vitreous right in this part. And here you have the retina, choroid, sclera, and the eyeball finishes here. So coming from, let's start with the retinal pigment epithelium. It provides nutrition to the photoreceptor cells in the front. The photoreceptors, they are nothing but the rods and cones. The rods enable the night vision, cones enable color vision and contrast vision. And then you have the bipolar cells, which are located here, which connects these photoreceptors and the ganglion cells. And finally, you have the ganglion cells, which are here. It transmits information from these photoreceptors to the brain through the optic nerve. The axons from these ganglion cells, they join together, form fibers, and they connect here, and finally forms the optic nerve, which leaves the eyeball and goes to the brain. So another important clinical aspect which you need to know is this image. Whenever you see this image in a fundus, the first thing you need to remember, especially in a country like India, is diabetic retinopathy, because the burden of diabetic retinopathy is increased a lot. And what happens in diabetic retinopathy? There's a damage to the blood vessels, which results in bleeding. You can see a lot of hemorrhagic bleeding spots around here. And the another clinical emergency is this condition, which is called retinal detachment. The patient usually complains of flashes, floaters, and defective field. It is a clinical emergency and immediate referral should be done. And the final and the last structure we have is the optic nerve. It is the second cranial nerve. It transmits visual impulses from the retina to the brain. The head of the optic nerve is called the optic disc. So how do you find out an optic nerve damage clinically? Watch out for something called RAPD. What is RAPD? RAPD is a relative afferent pupillary defect. So normally when you shine a torch on a pupil, the pupil should constrict. But what happens when there's an optic nerve damage? Instead of constricting, the pupil dilates. So that should point out, you should suspect an optic nerve damage, and you should immediately refer to a neuro-ophthalmologist. So that brings us to the end of the second presentation. We are going to end with just 2 questions. So question 5, which part of the retina has the maximum concentration of cone cells? So is it the optic disc? Is it the ora serrata? Is it the optic nerve? Or is it the fovea? Okay, 73% have got it right. It's the fovea. And coming to the last question. So the blind spot refers to which of the following structures? So this is the final question. Is it the optic disc? Is it the macula? Or is it the fovea? This must be a fairly simple question. So 64% have got it right. So to explain it, I have an image in the next slide. So this is the final image of this presentation. So this is an image of a fundus, a healthy fundus. So here, this right here is the optic disc, which is considered to be the blind spot because it does not have any photoreceptors. And right here towards the nasal side, you have the macula, and the center of the macula, you have the fovea. And this part is where you have the maximum concentration of cone cells. And this part is responsible majorly for your central vision and for your color vision and contrast sensitivity. With that, I thank Divya ma'am and Oro Siksha for giving me this opportunity. And that brings us to the end of the presentation.